So wonderful. Thank you, everybody, for joining us. So I'd like to get started with today's lecture at our single-cell spatial analysis program monthly seminar series. And it is really wonderful to welcome and introduce an absolutely exceptional and visionary scientist, Dr. Roel Verhaak. Dr. Verhaak is a professor in the Department of Neurosurgery at the Yale School of Medicine. And after graduating with a PhD in medicine from the Erasmus University Medical Center in Netherlands, Roel joined the Broad Institute of Harvard MIT and Dana-Farber Cancer Institute as a postdoc fellow. At the Broad Institute, he was on the team analyzing the data from the Cancer Genome Atlas TCGA, leading to the identification of the glioblastoma subtype, mesenchymal-like classical subtype. And since then, Roel moved to MD Anderson Cancer Center in Texas 2010 to start his own group. Since then, the Verhaak lab has studied tumor evolution in gliomas, and his group was foundational in establishing the GLASS consortium with primary and recurrent glioma patient sample, and established the recurrent resources of the model of tumor progression. And after being affiliated with the Jackson Laboratory for Genomic Medicine in 2016, his group moved to Yale last year. And lastly, I wanted to mention two things. One is that Dr. Verhaak is very visionary. His lab has studied extrachromosomal DNA amplifications, and this phenomenon with tiny DNA fragment was first discovered for a long time. But then, since then, it has been underappreciated. But along with Paul Michel, his group contributed greatly and recently received a lot of attention, for example, receiving $25 million of the support from the Cancer Grant Challenge. And now many people are rushing into the field. And also Dr. Verhaak has produced great scientists. They are very bright, sharp, and then deep knowledge, but also more importantly, they are good people. They're very generous and nice to the colleagues. So I think that this is rooted in the culture and science Dr. Verhaak has established. Accordingly, he has received numerous awards, including AAS Watcher Award, Adjunct Early Career Professor Award, Peter Steck Memorial Award, and he's a co-founder of the Boundless Bio. So again, it is great pleasure to welcome Dr. Verhaak for his seminar. Please, Dr. Verhaak.
Well, that's a very, very kind introduction. And now, of course, I have to live up to all those very nice things that you said. Let's see if I can share my screen. Yeah, I think that should look good, but let me know if not. So thank you again for the kind introduction, and thank you for the kind invitation, and it's my pleasure to speak to you today. The focus of today's talk is actually on IDH-mutant gliomas, and I'll introduce that a bit further. So IDH-mutant, so most people have heard of glioblastoma. It's the disease that took away Ted Kennedy. It's the disease that took away John McCain. And it's very much a devastating disease. It's also a disease that typically is found in patients that are 60 years or older of age. In contrast, the other type of glioma that we recognize is IDH-mutant glioma, which is frequently diagnosed as a low-grade glioma, suggesting that it's slow-growing. But these are also deadly tumors. Within the category of IDH-mutant glioma, we further recognize IDH-mutant gliomas with a 1p19q co-deletion, which are histologically mostly oligodendrogliomas, and then IDH-mutant glioma in absence of this 1p19q co-deletions, which are histologically mostly astrocytomas. Now, the oligos are typically diagnosed in patients that are about 45 years old, and the astros, or IDH-mutants without co-deletion, are typically diagnosed in patients that are 37. So that's much younger than the age at which most glioblastomas are diagnosed. And the associated survival with these two subtypes of glioma makes that actually you can't really speak of any kind of favorability here. And what I mean by that is that in all three cases, in all three types of glioma, IDH-wild type tumors, IDH-mutant with co-deletion, and IDH-mutant without co-deletion, the patient is looking at a significant loss of life, and often at age 60 or younger. So these are all truly devastating diseases. So I'm going to focus today on IDH-mutant gliomas. So these two tumor types, as I said, often also referred to as oligodendroglioma or astrocytoma, or in my talk, I'm going to use the acronyms IDH-O or IDH-A. These two tumor types are further recognized by very different sets of genomic alterations. So even though they share, let me see if I can get a pointer somewhere. Oh, yeah, there. Yeah. So even though they're universally mutated in the genes IDH1 or IDH2, so that they share that common mutation, you can, for example, find that oligodendrogliomas are also universally mutated in the TERT promoter, whereas these mutations are pretty infrequent in astrocytomas. In astrocytomas, you can find that 95% of cases at diagnosis contains a mutation in P53 and 75% is ATRX mutated, whereas these two events are pretty rare in oligodendrogliomas. So even though they share the IDH mutation, after that point of tumor initiation, which presumably the function of IDH, the next steps in tumorigenesis may be very different, at least as that is suggested by the differing sets of genomic alterations that are observed. So work by Mario Suva and Itai Tirosh has illuminated the different types of single cell states that can be found in this tumor type. And interestingly, when you evaluate the gene expression programs of IDH-mutant glioma cells and do a comparison between IDH-mutant oligos versus IDH-mutant astros, you find that they're actually at the single cell level not so very different. In fact, in both oligos and astros, there are three major cell states, starting from neural precursor cells, which are often cycling cells. And then afterwards, there's a hierarchy in which cells can either go towards the astrocyte-like states or the oligodendrocyte-like state. And the main difference between oligodendrogliomas and astrocytomas in this case is that the oligos lean more to the right side of the figure, the inverted Y plot here, and the astros lean more to the left side of the figure on this side. So despite the very different sets of genomic alterations, at the single cell phenotypic level, there's much similarity between oligodendrogliomas and astrocytomas. So intratumoral heterogeneity is driven by evolutionary processes and clonal selection. So at the time of diagnosis, every tumor in every part of the body actually shows intratumoral heterogeneity, sets of cells marked by the same, by particular sets of mutations. And this intratumoral heterogeneity is, of course, shaped by clonal selection processes, where specific mutations provide specific advantages in a tumor microenvironment. Now, this process of clonal selection and tumor evolution doesn't end after diagnosis. It continues. And in fact, when we treat the patient, encourage this effect, we contribute to the clonal selection first by debulking surgery, where we try to remove most of the tumor cells. Then by irradiating the remaining tumor cells, and then finally through chemotherapy. And only those cells that are not being removed through resection and that can survive the toxicity of radiation and the toxicity of chemotherapy, which is mostly temozolomide, only tumor cells that can survive all these pressures will eventually go on to give rise to the tumor recurrence. Now, as I showed in the introduction, the treatments we use for these patients are generally quite ineffective, which means that ultimately the tumor will always recur, and at each step of recurrence, the tumor is more refractory to further treatments. So if we want to understand how we can optimize the use of these treatments and maybe provide additional effective treatments, characterizing and delineating this process of treatment-induced tumor evolution may be quite important. And that actually formed the basis for the glioma longitudinal atlas, the GLASS consortium, where we do comparative sequencing of gliomas across their treatment trajectory. I'm actually not going to spend a whole amount much time on the GLASS consortium data, because I think we have built upon the GLASS consortium data, and that has provided us with new insights. One of those new insights from our GLASS analysis, however, is that chemoradiation has a pretty strong impact on the tumor genome in IDH-mutant glioma. For example, Flores, who is now an assistant professor at TGen, but during his time in the lab, which very much overlapped with Minji's time at the Jackson Laboratory, Flores and others did an analysis of the GLASS consortium data to show that a large proportion of IDH-mutant gliomas goes on to develop what is called the hypermutation phenotype in following treatment with temozolomide. What that means is that after temozolomide treatment, when we sequence tumors after temozolomide, a subset has a very high mutational frequency, often 10 or more mutations per megabase. And this is markedly different from those that do not carry hypermutation phenotype. In other words, we have pretty much a binary distribution. A tumor is very clearly either hypermutated or they are not hypermutated. And this hypermutation effect is contributed to the chemical process that temozolomide brings to the glioma genome. Then Emre, who also spent time in the lab, went on to show that following irradiation, there's a strong enrichment for the presence of CDKN2A homozygous deletions. That means that CDKN2A homozygous deletion is pretty rare in IDH-mutant gliomas at the time of diagnosis, but following irradiation, nearly a third of tumors will have acquired CDKN2A homozygous deletions, suggesting that this loss of P16 somehow is able to play a role in the response to radiation. And in a separate analysis, Emre also showed that in a subset of tumors that have undergone irradiation, a small deletion signature somewhat similar to hypermutation is acquired, which means that in a subset of tumors, there's a smattering of small deletions across the genome that we can clearly link to the exposure to radiation. So to enhance the title again, chemoradiation indeed has a strong effect in a subset of IDH-mutant gliomas and alters the glioma genome. I want to make a small sidestep here and focus on APOBEC signatures, because I believe that those are another, in my view, quite interesting finding from our analyses. So in conjunction with the loss of CDKN2A and in conjunction with the acquired small deletions, we also found, especially in the cases that acquire small deletions, we found an enrichment for the mutational signature SPS13. And SPS13 is a APOBEC signature. So this figure shows the relative enrichment pre and post treatment in the GLASS data sets. And as you can see, SPS13 is clearly marked. And when we performed a similar analysis on a data set from many cancer types provided by the Harvey Medical Foundation, we found that in many other cancer types, we also saw this enrichment of SPS13 following irradiation, a significant enrichment. So somehow, APOBEC activity may be linked to the response to radiation therapy. And when we looked more carefully at the GLASS data sets, splitting into the IDH-mutant tumors and IDH-wild type tumors, and separating mutations that are shared across time points versus those that are specific to the post-irradiation tumor, we found a significant increase in APOBEC motif mutations in the newly acquired mutations, and not so much in the mutations that are shared. And this was true for both IDH-mutant tumors and IDH-wild type tumors, implicating that the presence of APOBEC-derived mutations is specifically linked to at least the post-diagnosis evolution of the tumor, which is very much informed by the radiation therapy. And Perhaps not so surprising if you know that the presence of APOBEC mutations converges with the presence of CDKN2A homozygous deletions. They're different sides of the same coin, if you will. But with that in mind, we find that high APOBEC mutation burden also associates with worse patient outcomes corrected for various potential covariants. So what is APOBEC exactly? Maybe a brief refresher. So APOBEC proteins provide a defense mechanism against retroviruses that typically consist of single-stranded DNA that then gets, of course, reverse transcribed into cDNA prior to integration into the genome. So APOBEC provides a defense mechanism by actively mutating single-stranded DNA, mostly in the cytoplasm, although one of these multiple APOBEC proteins also translocates to the nucleus. But it's mostly active in the cytoplasm. And there it tries to mutate single-stranded DNA, which should naturally not occur. Only RNA viruses should typically have, or retroviruses should typically have single-stranded DNA. So it's a defense mechanism to mutate that single-stranded DNA and thereby resulting in the degradation of these nucleotides. Now, during the cancer process, there's there must be maybe as a result of genomic instability, there must be single-stranded DNA that's available for mutation, because when you analyze a large set of newly diagnosed tumors or untreated tumors, you see that in subsets there's actually a pretty high contribution of APOBEC mutations, with bladder cancers being perhaps the front runners. In fact, some of these bladder cancers, as you can see here, most of their mutations may have been contributed by the APOBEC proteins. Note that in glioblastoma and low-grade glioma in this pan-cancer analysis, there's virtually no APOBEC mutation activity. And again, this is measured in newly diagnosed tumors. Observing that following irradiation, we do find APOBEC mutations in gliomas, we went on in an analysis led by MD PhD student Bianca to evaluate the exact role of APOBEC proteins in the formation of mutations after radiation, in response to radiation. And Bianca started this analysis by knocking out two of the most prominent members. Actually, most of the interest in this APOBEC family has gone to APOBEC 3b. But because we found that APOBEC 3g was more highly expressed, we've considered knocking out both 3b and 3g in several glioma-derived neurosphere cell lines, then treating those cells with 5 doses of 2 grays, which is similar to the clinical regimen, and then comparing whole genome sequencing from baseline to whole genome sequencing after about 25 cell doublings to give cells adequate time to recover and incorporate new mutations. So Bianca performed this knockout experiment as well as the whole genome sequencing, and then analyzed the data with the question, do we see evidence of APOBEC signatures? And the answer is very much, first of all, yes. If you look at the scrambled control, irradiation indeed induces or increases the level of APOBEC mutations. And this is true for both point mutations as well as small insertions and deletions. So we see more of those following irradiation. However, if you knock out A3G, we actually are able to abrogate the activity. We do not find that increase in mutations, true for point mutations as well as small deletions. And that's not true for APOBEC 3b. So there's a clear divergence there, with APOBEC 3g being, at least in this analysis, the most relevant and most active APOBEC family member. Bianca then performed clonogenic assays to understand if knocking out APOBEC members could actually not just play a role in the, not play only a passive role, but actually somehow contribute to the response to DNA damage. And the answer appears to be yes, which is a little surprising perhaps. So knockout of A3G as well as A3B, at least in this Henry Ford cell line that we got from our collaborator, Anna De Carvalho. In this Henry Ford cell line, we found that knocking out both these APOBEC members actually reduced the cell viability, suggesting that this is actually potentially a radiosensitizing strategy. And in this second cell line, we continue to see a clear effect for knockout of A3G, less so for A3B. So again, enhancing the point that APOBEC 3G appears to be the most prominent APOBEC protein in response to radiation therapy in gliomas. Bianca then performed a rescue experiment where she then re-expresses wild-type APOBEC 3G or a catalytic domain inactive version of APOBEC, and in both cases finds that the re-expression actually restores the radiation response, further providing evidence that APOBEC directly plays a role in the response to radiation therapy. Finally, we used a APOBEC inhibitor, which I think is a very toxic drug, but still it's reported as APOBEC inhibitor, and find that across multiple dosages of this inhibitor, we see indeed an abrogation of the, an augmentation of the effect of radiation by reducing cell viability of the combined radiation and APOBEC inhibitor, although of course it should be noticed that the inhibitor itself already has a pretty strong toxic effect. So it's a pretty dirty drug. But summarizing this first part, and I specifically included this because Minji said that she appreciated the experimental work that the lab has been doing. Concluding this first part is that we think that there is indeed a role for APOBEC3 inhibition towards radiosensitization, and this is definitely an area that we're continuing to investigate. All right, so back to the data analysis. So in total, in our GLASS cohort, which now consists of well over 100 IDH mutants, we find that about 60% of IDH mutants following disease recurrence, we find evidence of a new genomic alteration that we think is important for tumor regenesis, including temozolomide-derived hypermutation, radiation-induced homozygous deletions, radiation-induced small deletions, and to some degree, radiation-induced aneuploidy. And as I said, in totality, about 60% of tumors acquires one or more of these alterations. And of course, of relevance is that in tumors that acquire such an alteration, survival is going to be significantly worse for those patients relative to patients whose tumors stay genomically relatively stable. And we think this is potentially an important observation. It also supports this model of clonal selection because it follows the idea that there's a cell of origin who gives rise to multiple subclones at the time of diagnosis, marked by different sets of mutations. After the therapeutic barrier, there are going to be some cells remaining that are able to repair the DNA damage induced by, in our case, temozolomide or irradiation. And those then fuel the disease recurrence. And we know that they fuel the disease recurrence because these have to be cancer clones that are large enough to be detected through sequencing, which I would say has generally a threshold of about 20% of tumor cells. So 20% of tumor cells need to carry the same mutation in order for it to be detected through standard whole genome sequencing. To further understand exactly how this process of tumor evolution unfolds, we struck up a collaboration with several labs, including those led by Mario Suva at MGH, Itai T. Roche at the Weizmann, as well as Antonio Iavarone and Ana Lesarella, who are now at the University of Miami. And this work was, what I'm going to talk about specifically was led by Kevin Johnson, but there were many contributors to this analysis, and there were many collaborators that we also recognize in this large effort. So in short, we identified 35 patients for whom we could obtain tumor samples from the initial tumor at diagnosis as well as the recurrence after that occurred after treatment. So in total, we obtained 75 tumor samples from 35 patients, which means that there were a couple of patients for whom we had actually 3 time point tumors or even more. And those 35 patients were distributed over the 2 IDH mutant subtypes. From this sample collection, we generated single-nucleus RNA-seq data for 27 patients. For the remainder of the 75, we generated single-nucleotide RNA-seq as well as single-nucleotide chromatin accessibility data, so single-nucleotide ATAC profiles. And for 90% of the cases, 95% of the cases, we generated either whole exome or whole genome data. And the focus of this analysis is on the single-nucleus data. So first, when you do your standard UMAP analysis of IDH oligodendrogliomas or astrocytomas, you get dot clouds such as these ones. When we then use marker genes to further characterize the tumor cells as well as the tumor microenvironment cells, we can deconvolute each tumor and the components that you would expect to find. First of all, a relatively high tumor purity. Second, many oligodendrocytes in the tumor microenvironment, and many myeloid cells in the immune compartment of these tumors. So Mario and Itai's earlier analysis had identified 3 cell state programs, but that analysis was based on a small set of samples with a older technology that captured fewer cells. When we repeated the efforts to identify malignant cell states, we were able to extract through computational methodology that's been pioneered by Itai and his group, 4 malignant cell states, and we labeled those cell states based on marker gene expression as OPC-like, AC-like, MES-like, and NPC-like. And then in parallel, we annotated each individual cell for whether they were cycling, yes or no. Now, if you have worked in the field of glioma recently, you have likely heard these cell state labels before. In fact, in Mario and Itai's famous paper from 2019, where they performed single-cell analysis of glioblastomas, he also found 4 cell states and labeled them also OPC-like, NPC-like, AC-like, or MES-like. So the cell states we have now derived with our updated analyses and our larger comprehensive data sets are actually highly reminiscent of IDH-wild type glioblastoma. And I actually think that's pretty remarkable that even though we see so many differences in these tumor types, differences in age, differences in marker gene expression, in genomic alteration, differences clinically in the way that these tumors respond to therapy. Despite all those differences, at the phenotypic single cell state level, there's actually a lot of similarity. Now we can take I should say that there's actually a subset of cells that are not neatly classified in any of the 4 buckets, or perhaps that is a difference between mutants and wild types. And we classified those tumor cells as undifferentiated. And they likely reside somewhere in between all of the particular cell states, because we could not infer a single characterizing signature from these undifferentiated cells. When we now project these single cell programs on the inverted y-axis, and as a brief reminder, the inverted y is more stem-like at the top and more differentiated at the bottom, with the left leg towards astrocytic, the differentiation, and the right axis more towards oligodendrocytic differentiation. When we now project these cell states on there, we see somewhat of a discrepancy between first NPC-like and undifferentiated cells, which are clearly more earlier in development, more stem-like, if you will, whereas MES-like and AC-like really perturb into the various differentiated axes. So we see some cell states that are clearly more stem-like, and we see other cell states that are clearly more differentiated. When we focus on the more stem-like cells, we then use different colors for the number of cycling cells, we see that, of course, as had Mario and Itai shown before, that most of the cycling cells reside at the top of the inverted Y, as expected. So the earlier cells are more likely to be actively cycling. And here I come back to that somewhat dichotomy of states with NPC-like and OPC-like, as well as undifferentiated cells, containing the majority of stem-like cells, of cycling cells, and cycling cells being relatively rare amongst the mesenchymal-like and AC-like cell populations. As we had generated simultaneous chromatin accessibility data, we could take the cell states derived from single-cell RNA sequencing and then use computational algorithms to infer master regulators of each of those cell states. And we come across names of gene names that we have observed before, such as PDGFRA, which of course is a marker for astrocytic development, OLIG1, which is a marker of oligodendrocytic development, aquaporin-4, also astrocytic, and so on and so forth. Yeah, this sort of makes the same point as I just made. And actually what we then, digesting the chromatin accessibility data into a single analysis, when we count enrichments across various types, we see that regulators of gliogenic switch are activated in a cell state dependent manner. So, for example, NF1B and ASCL1 over there are well known to direct early progenitor cells into either of the two astrocytic or oligodendrocytic lineages. And these are also the same type of regulators we found associated with these glioma cell states. And if you put this in a bigger scale, actually, this really starts to be reminiscent of hematopoietic development, where it's well known that different leukemias are stuck in a particular cellular differentiation state. And if you were able to push the cells out of that differentiation state, you actually have the ability to have a treatment for those patients. And that's what I think we're starting to see for glioma as well. More importantly, we found that grade increases converge with dedifferentiation and increased proliferation. That's perhaps an observation we could have made by talking to a pathologist who will argue that this is, of course, the case, that a recurrent higher grade glioma has signs, all the signs of dedifferentiation and higher levels of, for example, Ki67 staining. And we found this for both IDH-mutant oligos as well as astros. However, when we put that data together, comparing not tumors by grade, but by time point, we see similar shifts in that across 35 tumors, we see a significant decrease in the number a significant increase in the number of AC-like cells and the number of MES-like cells, as well as the number of cycling cells. When we do a paired analysis projecting the median point of each of the tumors and linking the time point tumors, we see that most tumors indeed shift upwards, meaning that their average level of differentiation transitions to an earlier state. Now, here's perhaps what I think is the most impactful finding of this study. When we now take our set of IDH-mutant gliomas and separate them into two groups. The first group being samples where we see evidence of a therapy-induced genetic alteration, meaning hypermutation, loss of CDKN2A, or a large increase in the number of copy number alterations. And about 60% of our cohort falls into this category. And then we take the other group of samples where we do not find that evidence for therapy-induced genetic alterations. What we observe is that the cell state changes almost exclusively occur in the first group, meaning there's a very strong convergence between acquiring a genomic alteration as a result of treatment, and cell states transitioning to becoming more earlier away from the more differentiated AC-like states towards a higher rate of cycling cells, towards more undifferentiated cells. The only notable exception here is that across both categories, we see an increase in the number of mesenchymal cells. Now, 35 tumors is not a huge data set, but fortunately, we did have access to the GLASS data sets where we don't have, per se, single-nucleus RNA-seq, but we have bulk RNA-seq that we can deconvolute computationally into different cell types. And when we repeat this analysis, first of all, we recapitulate the fact that we see generally de-differentiation and trends towards higher rates of cycling. And again, when we split the bulk RNA data set from GLASS into a cohort of samples with evidence for therapy-induced genetic alterations, and a second cohort for samples without therapy-induced genetic alterations, we pretty much recapitulate the same observations, meaning there's a decrease in AC-like state, there's an increase in NPC-like state, there's an increase in undifferentiated cells. We were not able to recapture the increase in mesenchymal-like cells, but we actually think this is a limitation of the bulk RNA-seq platform that is not quite sensitive enough to capture the mesenchymal-like signature. As I pointed out now several times, we see an increase in the mesenchymal-like cell state at tumor recurrence. We see this in both the single-cell data as well as in the bulk RNA-seq data. And we find that similar to what has previously been described actually by Toshi and others, we find that the increase in mesenchymal-like state in IDH-mutant gliomas correlates with an increase in myeloid cells in the tumor microenvironment, namely right there. Perhaps not a huge surprise, but still interesting to capture for IDH-mutant gliomas. Finally, I want to show how you can reanalyze single-cell data to study genomic events. As I pointed out before, temozolomide is a dealkylating agent, and through the dealkylating process, a C gets changed to a T, and as a result, you get a point mutation. And of course, point mutations we can pick up through exome or whole genome sequencing. However, single-cell sequencing data has ability to also identify such changes. It's just that the data from single-cell sequencing, of course, is very sparse. On average, at least in our case, we get about 50,000 reads per single cell. So 50,000 reads per single cell is not sufficient to cover, let's say, a p53 mutation. But you can certainly analyze the data sets to identify any kind of mutation. And when we take that approach, we can enumerate all the mutations we can detect in a single cell and evaluate whether those mutations are the type of C to T change that is associated with temozolomide treatment. And when we take that approach using our single-nucleus RNA-seq data set, and we split our tumors by whether they are hypermutated or non-hypermutated, we see that this approach indeed can recover a subset of single cells where the tumor genomes have undergone this hypermutation event. We can very clearly see this dichotomy into cells with a high C to T contribution and cells with an absence or a very low C to T contribution. And because a subset of tumors in our data set had simultaneous single-nucleus RNA-seq and single-nucleus ATAC-seq data, we can validate this observation in the single-nucleus ATAC-seq profiles. So with single-nucleus RNA sequencing or ATAC sequencing, you can classify, you can bin cells as to whether they are hypermutated or non-hypermutated. Now, we've taken that approach for the 7 tumors that have undergone hypermutation and split the single cells from those 7 tumors into C2T low or C2T high to understand the patterns associated, single cell state patterns associated with hypermutation. And when we take that approach, we see that the non-hypermutated cells are significantly increased in terms of the number of more differentiated AC-like cells. the hypermutated single cells are more undifferentiated, more early in their differentiation process. And even though it's an insignificant trend, we see a trend towards a higher rate of cycling in the hypermutated cells. And when we project the hypermutated versus the non-hypermutated cells and the inverted Y, we also can show a significant difference. Finally, we can try and classify single cells by amplification status. For example, for the gene PDGF array, which for astrocytoma has been associated with relatively poor outcomes. We can use our parallel whole genome sequencing data to define specifically a boundary for a focal amplification, and then use a change point analysis to infer which tumor cells maintain that focal amplification at the single cell level. So we took the 5 tumors in our cohort that contained a PDGF array amplification and separated those by ATAC-seq data into whether the sample, whether the cell was PDGF array amplified, yes or no. And as you can see, this works fairly well because we can look at the orthogonal PDGF array expression levels to find that that's significantly higher in the cells classified as containing the PDGF array amplification. And then we can perform a similar analysis of the previous that I showed for the previous for the hypermutation example, and to show that, for example, PDGF array amplified tumors are significantly higher in terms of their cycling abundance, and significantly lower in their level of differentiation. So then that leads me to just about to wrap up here. I think what we're now showing is that, first of all, tumor evolution and clonal selection continues with the therapeutic barrier. We have previously hypothesized and provided evidence that cancer cells able to repair DNA damage, such as hypermutant cells, are the ones that give rise to the recurrent tumor. They're directly driving the recurrent tumor formation. And through our single-cell analyses now, we actually show that this is the case because we clearly see that it's the hypermutant samples and hypermutant single cells that give rise to the earlier phenotypes and the higher rates of cycling. So to conclude, IDH-mutant cell states are very reminiscent of those that have previously been described for IDH-wild type glioma. IDH-mutant glioma progression is largely driven by acquired genetic alterations because we clearly see that difference in outcomes and sensitivity to treatment between tumors with and without acquired genetic alterations. These acquired genetic alterations drive dedifferentiation programs and higher rates of cell cycling. And then finally, we have recovered a secondary axis driving disease recurrence in a shift towards a mesenchymal-like state, which converges with an influx of myeloid cells, which of course has been described for IDH wild-type gliomas, for example, in association with the sort of cell cooking effect of radiation and the associated hypoxia that arises. So with that, I'll conclude, and I would like to, of course, thank all the lab members. I've recognized those that have led the various projects along the way, and happy to take questions. Thank you.
Thank you so much, Dr. Verhaak. It's a wonderful work. So the floor is open for having multiple questions. Maybe I can go first. So you observe the mutual exclusivity between CDK4 homozygous deletions and macrophages or myeloid cell recruitment. Do you know any evidence that the deletion of the CDK4 can basically suppress the myeloid cell recruitment or the induction or the mesenchymal program in the tumor?
You mean the CDKN2A deletion?
Yes.
Okay. I actually think it may be almost the reverse. I think the presence of CDKN2A deletions is likely to converge with the presence of myeloid cells.
Presence of myeloid cells.
Yeah, because we find it after radiation therapy, and we have also previously found that after radiation therapy, you see an influx of myeloid cells. Basically, the way I picture it is that after radiation, it's basically an inflamed mess in there. So you get an influx of macrophages to try and clean up all the debris that has been created by the irradiation process.
Thank you so much. Dan, please.
Yeah, Dr. Verhaak, I really enjoyed it. I'm Dan. I'm a radiation oncologist. I'm so glad you're thinking about radiation and recurrence afterwards. I had a question about the ApoBec part of the story. Do you think that this is important for the repair of like the single strand lesions that occur after radiation? And do you think it's more important in the IDH wild type mutant cells or both?
That's a great question. And radiation is so interesting in that we see so many effects of radiation on the tumor genome in our GLASS analyses, for example, and we see the same changes in genomics actually in other tumor types too. So I'm all with you in thinking about radiation. So I think the short-term effects are similar between IDH-mutant and IDH-wild type gliomas. It's just that in IDH mutants, there's more time for the clones in which these effects have begun to grow out and become the major clone in a particular tumor. Whereas in IDH wild type tumors, there's many cells that cycle quickly, and therefore there's not as much chance for a competitive process to fully unfold, and therefore we may not find it as clearly as we see the IDH mutant gliomas, but I think they're important in both. And then the exact mechanism by which ApoBec contributes, I don't think we have clear evidence for any particular mechanism. I mean, just from literature review, we can see that ApoBec is important for single-stranded DNA mutagenesis. What I could picture is some scenario where the activity of ApoBec can enhance the DNA repair process, and that it's some sort of bystander effect, unintended, but actually beneficial to the tumor cell. And that may be why we are also detecting it, because if it wasn't beneficial, the ApoBec mutant cells wouldn't be clonally selected. So I think without any kind of proof or evidence for this hypothesis, I would speculate that maybe it's an unintended effect and not a direct effect that ApoBec directly is able to contribute to DNA repair.
Thank you.
Thank you so much. I have another questions, for example, related to the ApoBec work. So what is the actual inducer or activator of the ApoBec upon the treatment with the radiations? And also, do you see any evidence that normal cell in the tumor microenvironment may be affected by the irradiation to acquire certain sort of mutations or other effect?
So that second question is very interesting, but we have really not done any analyses there. But it's certainly, it seems very logical that normal cells would also be affected. However, with genomics, as you know, you're going to detect any kind of changes that are present in the majority of cells, or at least enough cells that you can detect it through sequencing. And that only occurs if you have clonal selection, right? And I don't know that there would be such strong clonal selection for cells.
True.
So
Yeah.
it may be there, but you may need single cell approaches, which of course are very costly at the single cell level. If you had to do a single cell whole genome sequencing or something to detect those changes, but I think they probably are there. We probably can't detect them. Sorry, your first question on ApoBec?
Is the activator of the ApoBec or the expression level of the ApoBec is increased upon the irradiations?
Actually, it's a great question. I actually took out that slide, but yeah, radiation increases at least in our in vitro, in our neurosphere models, radiation induces the expression or increases the expression of ApoBec proteins, in particular ApoBec 3G. And that's another reason that I didn't show, but that's another reason why we're mostly focused on A3G in our analysis.
I see. So then maybe the lower expression level at the basal level, we don't see ApoBec signature in glioma, but other cancer type, they have higher expression of ApoBec. and then hard circumvention
Yeah, it's
based on
possible.
the expression.
Yeah, we don't really quite have enough data, I think, to show that, but it's a reasonable hypothesis.
Sounds good. Thank you. Pedro, please.
Hi, Pedro Lowenstein here. Great talk. Two related questions to the IDH1 mutation. You say that 60% of the IDH1 mutated tumors have an increase in the mutation rate. Can you speculate what causes the progression in the other group?
Yeah, that's of course a very good question, and nice to see you, Pedro. So we see the increase in, for example, the mesenchymal-like tumor cell state. It's hard to know what causes the formation of these mesenchymal-like cells, but in general, those cells tend to be more resistant to any kinds of therapies. So that could be one response, is that the mesenchymal cells are driving the tumor progression for the tumors that do not acquire genetic alterations. I think even if you take that away, there will still be tumors that are progressing in absence of mesenchymal cells, and exactly why those are progressing, I don't have a good response to that. But I would speculate that those tumors where you don't see acquired genetic alterations continue to be sensitive to treatment. So even though they're progressing, they will continue to have a treatment response. It's my speculation. Be interesting to try and test that.
Do you know if pathologists can differentiate those two groups?
Yeah, we're not quite there yet. Let us publish this paper first, and then we'll engage with pathologists to try and train models to do that. That would be a wonderful, if we could, that would be actually really interesting.
Okay, thank you.
It's a great suggestion.
Roel, this is Shriram. Really beautiful talk. Really enjoyed the depth and breadth of what you presented. So I was just wondering, since IDH inhibitors are very much in the news and their effects on gliomas, and recent work from Mario's lab showing that there's a differentiation shift in the oligodendral glioma cases. Do you think that pre-treatment, post-treatment, based on the differentiation status changes that you showed, will respond differently to IDH inhibitors?
Yeah, it'd be great to analyze, right? We're waiting for those tumor specimens to materialize, right? Over time, we're going to, of course, see patients that have been treated with IDH inhibitors. And once we have a couple, we'll start sequencing them, and then we can try and address those questions. Mario's paper is the first in that sense. And they actually seem to show a trend towards differentiation, right? So it's almost
towards an AC-like.
Yeah. It will be interesting to get data from patients who have received multiple treatments, and ultimately trying to understand if maybe they need to be given in parallel, and that actually is killing both the differentiated and undifferentiated cells or something like that. But yeah, it's too early to answer.
Okay.
That'll be very interesting. And over time, we will answer the question.
Thank you.
All right, Roel, very nice talk. I appreciate you sharing the experimental and computational component of your lab. I think Toshi mentioned earlier in your introduction that you also pioneered the world of extrachromosomal DNA. So do you want to briefly explain to us how ecDNA fits into your story?
Sure. Well, actually, in the IDH-mutant glioma, ecDNA is not very common. I think some of the PDGFRA amplifications I talked about at the end may be extrachromosomal, although I don't know. I don't remember if we looked at whether they were chromosomal or extrachromosomal. However, ecDNA or extrachromosomal DNA is very common in IDH-wild type gliomas, and that's why actually we were relatively early to find them, because we were studying the genomics of IDH-wild type gliomas for a long time, and we continue to do that. So I don't think there's a big place for ecDNA in this particular analysis, in this particular story where we looked at IDH mutants, but ecDNA I think is potentially very important for the IDH-wild type tumors. We are in a different part of the lab with Inhi, who you know very well, who's now at Michigan State, trying to figure out how we could actually try and target ecDNA as the vehicle for oncogene amplification, rather than trying to target the oncogene itself. But that's future work that's not quite at the clinical level yet.
Great. We'll invite you to Ann Arbor in person next time at my department.
Be wonderful. And frankly, I was invited to go in person, but we couldn't work out the timing for that, so we decided to do virtual this time. be happy to come back.
Thank you.
Exciting. Yeah, we will definitely look forward to see you in person. So if there is no additional questions, and then we want to close today's seminar. Once again, I really appreciate your presence, Dr. Verhaak. We really appreciate your exciting works. Thank you so much. And also thank you so much, everybody, for your attendance.
Yep, thanks for having me. Take care, bye-bye.
Thanks so much. Have a good day, everybody.